Human ancestry and DNA analysis provide valuable insights into our origins and evolutionary history. By studying genetic markers and conducting DNA analysis, scientists have been able to unravel the complex web of human ancestry, migration patterns, and genetic relationships. Here's a deeper exploration of human ancestry and DNA analysis. Genetic markers. DNA contains genetic information that is inherited from our ancestors. Genetic markers, such as single nucleotide polymorphisms, SNPs, and short tandem repeats, STRs, are specific regions in the DNA that show variation between individuals and populations. By analyzing these genetic markers, scientists can trace and compare the genetic patterns among different populations. Mitochondrial DNA, MTNA. Mitochondrial DNA is inherited maternally and provides insights into maternal ancestry. By analyzing the MTNA sequences, scientists can trace back the maternal lineage of individuals and identify common ancestors. Mitochondrial Eve is the most recent common ancestor of all living humans, traced through maternal lineage. Y-chromosomal DNA. Y-chromosomal DNA is inherited paternally and allows the study of paternal lineage. By analyzing the Y-chromosomal DNA, researchers can trace the male lineage and identify common ancestors. The term Y-chromosomal atom refers to the most recent common ancestor of all living humans traced through paternal lineage. Genetic variation and diversity. DNA analysis reveals genetic variation and diversity among human populations. Certain genetic markers or variations are more prevalent in specific populations, providing clues about historical migrations, genetic admixture, and population movements. These genetic markers can be used to map population histories and identify ancestral connections. Population genetics. Population genetics investigates the distribution of genetic variation within and among populations. By analyzing genetic data from diverse populations, scientists can study the genetic structure, gene flow, and evolutionary relationships between different groups. This helps in understanding human migration patterns, population interactions, and the formation of distinct populations. Human migration. DNA analysis has shed light on human migration patterns throughout history. By comparing genetic data from different populations, researchers have been able to trace the movements of ancient populations and identify migration routes. For example, the genetic analysis of Native American populations has provided insights into the peopling of the Americas and the migration routes taken by early human populations. Deep ancestry. DNA analysis can provide information about deep ancestry and ancient human populations. By studying ancient DNA from archaeological remains, researchers have discovered previously unknown human species, for example, Denisovan, and identified genetic admixture events between different hominin groups. This has led to a better understanding of human evolution and the complex interplay between different hominin populations. Genetic health traits. DNA analysis also offers insights into genetic health traits and predispositions to certain diseases. By studying genetic variations associated with diseases, researchers can understand the genetic basis of specific conditions and develop personalized approaches to healthcare. DNA analysis has revolutionized our understanding of human ancestry, migration, and evolution. By studying genetic markers and analyzing ancient and modern DNA, scientists continue to uncover valuable clues about our origins and genetic heritage. This knowledge not only enhances our understanding of human history, but also contributes to fields such as anthropology, archaeology, population genetics, and personalized medicine. Who were Denisovan? Denisovan were an ancient group of hominins who lived in Asia tens of thousands of years ago. They are known primarily through genetic analysis of a single fossil finger bone and a few teeth discovered in the Denisova cave in the Altai Mountains of Siberia, Russia. Here's what we know about Denisovan. Discovery. The existence of Denisovan came to light in 2010. When scientists sequenced the DNA from a tiny fragment of a finger bone, known as the Denisova 3 or Sha finger bone, found in the Denisova cave. The bone fragment was estimated to be at least 50,000 years old. Genetic analysis. DNA extracted from the Denisovan finger bone showed distinct genetic differences from both Neanderthals and modern humans. The analysis revealed that Denisovan were a distinct hominin group, genetically separate from Neanderthals and modern humans. Genetic interactions. The analysis of modern human DNA from various populations around the world has shown that certain populations, particularly those in Asia and Oceania, have traces of Denisovan DNA in their genomes. This suggests that interbreeding occurred between Denisovan and early modern humans. Geographic range. While the Denisova cave is located in Siberia, the genetic evidence indicates that Denisovan had a broader geographic range. Traces of Denisovan DNA have been found in modern human populations from Southeast Asia, Melanesia, and Australia, indicating that Denisovan were present in these regions in the past. Genetic contributions. The interbreeding between Denisovan and early modern humans has left a lasting genetic impact. Modern populations in Asia and Oceania carry small but significant amounts of Denisovan DNA in their genomes. This genetic inheritance has provided some populations with unique genetic adaptations and traits. Appearance and behavior. As of now, very little is known about the physical appearance or specific behavioral characteristics of Denisovan due to the limited fossil evidence. However, genetic studies suggest that Denisovan had adaptations to high-altitude environments, possibly indicating a degree of environmental specialization. Despite the scarcity of fossil remains, the discovery of Denisovan has significantly expanded our understanding of the complexity of human evolutionary history. It highlights the intermingling and interbreeding between different hominin groups, adding further intricacy to the story of human origins and genetic diversity. Ongoing research and future discoveries may provide more insights into the precise nature and significance of Denisovan in the broader context of human evolution. What was the reasons of their extinction? The reasons for the extinction of Denisovan are not yet fully understood. Due to the limited fossil evidence available, it is challenging to determine the exact circumstances that led to their disappearance. However, there are several theories and factors that may have contributed to their extinction. Limited population size. One possibility is that Denisovan had a relatively small population size, which made them more susceptible to factors such as genetic drift, reduced genetic diversity, and increased vulnerability to environmental changes or competition with other hominin groups. Competition with modern humans. The interbreeding between Denisovan and early modern humans suggests that there was overlap and potential competition for resources and habitats. As modern humans expanded and migrated across different regions, they may have outcompeted or assimilated the Denisovan population. Environmental changes. Changes in the environment, including climate shifts and the availability of resources, could have had an impact on the survival and adaptability of Denisovan. If they were highly specialized or limited in their ecological niche, changes in their habitat or food sources could have had detrimental effects on their population. Disease and epidemics. Like any species, Denisovan may have been susceptible to diseases or epidemics, which could have significantly impacted their population. Infectious diseases transmitted by modern humans, to whom Denisovan were closely related, could have had devastating consequences for their survival. It is important to note that our knowledge of Denisovan is still limited, and ongoing research and discoveries may provide further insights into their extinction. By studying ancient DNA, analyzing more fossils, and gaining a deeper understanding of the environments in which they lived, scientists hope to unravel more details about the factors that led to the demise of the Denisovan population.